Alright, so here's my presentation on atherosclerosis. So this is pretty much my interpretation of it before I actually really read into it a lot. Um, so essentially, when I think of atherosclerosis, I think of fat deposits of plaque that clog our arteries, which obviously causes narrowing, which reduces the blood flow and oxygen delivery. So when I did a little bit more research, this is kind of what I found, and after reading through all the articles on atherosclerosis, I'm realizing that it's very quite complex. So essentially what it is, it's a metabolic syndrome, and it can be caused by a variety of things such as obesity or unhealthy lifestyle, or a defect in adipose tissue, and the list just kind of goes on. Now, if we're talking about the liver, which is considered our biochemical factory, if that goes down or becomes faulty, then that can lead to major cardiopulmonary risks. So if we have a lot of fat accumulation in the liver, that is considered a marker for hepatic insulin resistance, which correlates to a lot of components of the metabolic syndrome that is separate to obesity. A lot of the studies suggest that a fatty liver can determine metabolic syndrome and cardiovascular disease, and inflammation of adipose tissue and a fatty liver seem to coincide, and they can be reduced by weight loss and PPAR agonist, which stands for peroxisome proliferator activated receptor. The effects of diet and exercise on liver fat needs to be studied more, and genetic factors also take part in the discussion as well, but there's a rise in liver disease associated with metabolic syndrome, so we need to address these questions and studies sooner than later. The adipose tissue plays a big role in insulin resistance that we see in patients who are obese and or have metabolic syndrome. The expansion of these cells in obese patients cause a pro-inflammatory state in the tissue, and macrophages begin to infiltrate. The increased amount of cytokines in tissue, and kind of like some of them I wrote up there, like TNF, interleukin 6, interleukin 8, MCP1. Create a pro-inflammatory state, impair the normal differentiation of the pre-adipocytes, and alter the pattern of secreted adipokines by the adipose tissue, and create insulin resistance, which will enhance the pro-inflammatory state in the tissue because the insulin causes that anti-inflammatory effect. An important thing to really remember and know is that the metabolic syndrome has to do with the dysregulated adipose tissue, and we get this adipose cell enlargement and infiltration of macrophages, like I mentioned earlier. The enlargement causes a pro-inflammatory state in the cells with less secretion of adiponectin and more secretion of cytokines and chemokines like interleukin 6 and 8 and MCP1, which plays a big part in recruiting macrophages into the adipose tissue. The more release of cytokines causes diminished differentiation of the pre-adipocytes, with less lipid accrual and stimulation of adiponectin, which enhances ectopic lipid storage. The TNF, the tumor necrosis factor, and interleukin 6 causes these effects in the pre-adipocytes, and this is related to a rise in WNT signaling, maintaining the cells in an undifferentiated pro-inflammatory state. And that will cause insulin resistance and a compromised inhibitory effect of insulin on FFA release, and the resistance really helps the pro-inflammatory state because insulin by itself is a anti-lipolytic and anti-inflammatory by antagonizing the cytokine-induced activation of STAT signaling. So for my next slide, as far as most interesting pathogenesis of atherosclerosis, I really kind of focus on VCAM1. VCAM1 is an immunoglobulin-like adhesion molecule expressed on activated endothelial cells, and it binds to alpha-4 beta-1 integrin, which is expressed on lymphocytes, monocytes, and eosinophils. The study shows VCAM1 is not expressed under baseline conditions, but it gets quickly enhanced by the pro-atherosclerotic conditions in animals and humans. Studies with cytokine-activated cultured endothelial cells and reconstruction assays with purified recombinant VCAM1 protein suggested that the VCAM1 could mediate robust adhesion of alpha 4 beta 1 expressing cells. Some evidence shows a pathogenic role of VCAM1 by showing that recruitment of peritoneal macrophages into the atherosclerotic lesion of mice can be inhibited by an alpha 4 blocking antibody and giving a small peptide that inhibits the ligand binding of alpha 4 beta 1, which delays the atherosclerosis in the mice. Some other studies that I read used using perfused carotid arteries harvested from mice, showing that between alpha 4 beta 1 and VCAM1 can slow down the rolling monocytes, and that VCAM1 promotes the monocyte adhesion and accumulation on the vessel wall at sites that are prone to developing atherosclerotic lesions. Another study I found talks about how VCAM1 is critical for the development of atherosclerotic lesions in an LDL receptor knockout mouse model, which is similar to human atherosclerosis and develops significant lesions when fed a pro-atherogenic diet. To increase the number of the surviving VCAM1, they removed just one of the two ligand binding sites of VCAM1, which caused a mutation in D4D, and this mutant lacks domain 4 of VCAM1, and its expression is much lower than that of a wild-type VCAM1. But it provides enough function to allow some of the homozygous mutant mice to survive to birth. And the amount of protection from atherosclerosis in the VCAM1 D4D mice was stronger than other mutants tested in the LDL receptor knockout model, which means that the VCAM1 is very important gene product that is very highly involved in making atherosclerotic lesions. In this picture that I drew, it kind of shows the inflammatory cells involved in atherosclerosis. So the inflammation in the fatty streaks. So after VCAM1 induction on the ECs, leukocytes, which I kind of wrote up there, the monocytes, T cells, and MCs enter the arterial intima by diapetitis, and differentiated macrophages will accumulate the modified lipoprotein or the OX-LDL by the scavenger receptors. And I don't know if I put the scavenger receptors, but it should be by the macrophage in that picture. But by the scavenger receptors, and they're converted into the foam cells. The T cells then direct their adaptive immune response in parallel. And the MCs release the granule components, which kind of enhance the endothelial permeability to plasma lipoproteins and inhibit the cholesterol efflux in the macrophages. The ICAM1, intracellular adhesion molecule 1, LFA1, lymphocyte function associated antigen 1, and VLA4, very late antigen 4. So now, for my fruit, I pretty much focused, I wrote all the things for fruit that I found and their roles in cardiovascular disease and how they can help, and they're very beneficial. My main focus, though, or the fruit that I picked, was blueberries. So overall, diet does play a huge role in preventing cardiovascular disease. And a study I read talks about how a diet low in fruits is the third most important risk factor for cardiovascular disease. I'm actually going to refer to cardiovascular disease as CVD, just to make it a little bit easier. And CVD following hypertension and cigarette smoking, actually. So quite a few fruits like grape, blueberry, pomegranate, apple, avocados all have been studied, and they all show to be very beneficial when it comes to preventing CVDs. Fruits have antioxidant and free radical scavenging properties, which are considered principal. And in these studies, they paid more attention to natural products rich in polyphenols, which is a group of compounds that has an aromatic ring, and phenolic hydroxyl groups. Polyphenols are acquired by diet because they cannot be synthesized or stored in the human body. And fruit is one of the main dietary sources of polyphenols. The richest sources of fruit polyphenols are actually dark berries. like grapes and blueberries. And some studies suggest that fruit-rich and polyphenols help control the cardiovascular diseases. But as far as blueberries goes, um, the cardioprotective effects of blueberries extract in the cholesterolomic rats is the study that I actually found, um, and it showed that blueberry extract decreased the aortic lesions. It lowered the serum lipid profiles and increased the activities of the antioxidant enzymes. The study showed that blueberry supplementation lowered systolic blood pressure by nearly 15% actually, and improved endothelial dysfunction and aorta relaxation in response to acetylcholine. I also found another study that evaluated possible protective effects of some phenolic acids that are identified as metabolites of blueberry on murine macrophage cells, and showed that phenolic acids decrease foam cell formation induced by the OXLDL bind to macrophages. The lipopolysaccharide induced mRNA expression and protein levels of TNF alpha and interleukin 6 by inhibiting the phosphorylation of mitogen activated protein kinase, which is MAPK. There's also JNK, which is June and terminal kinase, and P38 and ERK12. Downregulated the mRNA expression and protein levels of the scavenger receptor CD36 and upregulated the mRNA expression and protein levels of ATP binding cassette transporter A1, which is facilitated cholesterol efflux and inhibited cholesterol accumulation in macrophages. So overall, blueberries have anti-atherogenic properties, anti-inflammation, lower your blood pressure, improving oxidative parameters and vascular reactivity. And fruit in general will protect the endothelial function, decrease your blood lipids, decrease your blood pressure, alleviate ischemia, reperfusion injury, and inhibit thrombosis. And that's pretty much it, and these are all my references. Thanks.